So it's been my pleasure to work with Professor Erminio Monteleone here in Florence for the last 2 years, and he's going to be our last speaker for the morning. He'll be known to most, if not all of you, I'm sure, as a preeminent sensory scientist in Italy. Now, he's going to talk about, he's given his title in Italian, più di una semplice questione di gusto, sensualità e comportamenti alimentari. He'll explain to you what that means, I'm sure.
Thank you, John, and sorry. Grazie.
Thank you to the Bonduelle Foundation for organizing this day's meeting. For us, it's very important. And I, of course, must thank the University of Florence for the hospitality it has given to this day's meeting, and particularly to Professor Elisabetta Cerbai, who made this day possible. Then allow me to thank in advance. Well, today I'm talking as a teacher at this university and as the president of the Italian Association of Sensory Sciences. There are many people from the SIS present here today. Part of my presentation is dedicated to an important project that the SIS has decided to conduct. So I'd like to briefly greet my students, the undergraduates from the faculty and the doctoral students. So I thank all of you for being here. We've seen a view of presentations, very important, with very different viewpoints. My role will be to briefly, in the most simple way possible, to try to explain the potential role of the individual differences in perceiving sensory perceptions, in determining preferences and dietary behaviors. So we will be tackling a theme that is not totally new in scientific research. It has already achieved some consolidated results, and it needs to grow more and to make further progress. I will do this in a very simple way, starting from my experience as a researcher. I've been working in this sector of sensory perception for the last 20-25 years. My career has been looking at the fundamental phases. It could be confused with that of a schizophrenic. I've changed from one theme to another in the course of my career. In particular, I started by studying the chemical composition of food products and trying to understand their sensory properties and their relations. And then I moved on to the understanding the individual differences of individuals in perceiving sensory stimuli. Why did I do this? It's because I believe that the differences between people are more important than the differences between products for many reasons. In order to understand the differences between peoples, it's more important than studying products, because you can better identify products and better organize and plan policies for better nourishment in terms of health and individual well-being. Why is it important to understand sensory properties? The relations that we have to the flavor of food products, they're important to evaluate liking for certain food products. And very simply, the liking is associated with the quantities of a product consumed. And so this liking is a motivation for consumption, and it's a determinant for the mechanisms of preferences and choices. And when we study how much a food product is liked, we are used to looking at the average of the scores that we achieve. And when we look at these averages, we determine trends of acceptability, or which is greater or lesser, according to which we make our considerations, knowing that these judgments are important for consumption. But it's a more complex operation. The availability to investigate the individual's preferences, there are people who don't respond to sensory stimuli in the same way, and these differences are very deep, but they're little known, and that's our problem. Here you can see that, looking at those bars that represent the mean liking of a series of products, you can see these little dots, each of which represents a consumer, and his position on the map, on the chart, shows its position of greatest liking for that individual, that consumer. It's possible to identify the order of liking for that person, for that product he's evaluated. So this person J, who's got a trend, a pattern of liking, which is very different from the average one, and I could give examples on the same chart with other individuals who have reverse patterns of liking. So our need is to understand where the differences lie, what they're due to, and what are the fundamental facts that cause these differences. Here you can see an example according to the same logic of studying the differences between individuals individually. You can see each individual has a direction of liking, as these are individuals from 4 different countries, Italy, the UK, France and Denmark, represented here by different coloured triangles and squares, and these correspond to their liking for different types of vegetables. And you can see the concentration of forms such as triangles of the same colour, for example, in one portion of that chart. But if you look carefully, within each group, if you look at the white squares or the grey triangles, you can see a difference within the same group, which is huge. And this means that when we look at the mean differences, we really see very little, and some mechanisms of these mechanisms are escaping us in relationships with products. We see the different preferences and different likings are related to consumption and eating behaviors, as we've seen in the previous presentation. You can understand that if we do not enlarge and extend our way of exploring the reasons for these differences, we will still be chasing after problems which are not easy to solve. In 1970, Professor Pankborn, who all the experts in sensory valuation know, highlighted that if you compare the ways of reacting in hedonic ways to the concentration of saccharose, the response within the same population, the American population in this case, couldn't be summed up in one form of response. Pankborn found 3 different clusters of subjects who were diversified for their hedonic reaction to saccharose. So we are here looking at the results of experiments showing that food preferences are the result of these two fundamental facts. One is the environment, including exposure to products and experience of consumption of products. And on the other hand, there's the thrust of the biological factor, that is, genetic information which is transformed into behavior. Somehow it interferes or interacts with the mechanisms of exposure and experience, giving rise to individual differences in preferences. Strangely enough, though, when we see what people study in order to have this consumer segmentation, we don't consider the sensory stimuli or the differences in liking expressed for real stimuli. More frequently, we relate to, and more widely, we refer to the parameters that lead to determining preferences in terms of socio-demographics, socio-cultural characteristics, food behaviors, food lifestyle, attitudes, motives, and values. All of these factors are further reasons for segmentation between individuals when trying to decide on likings, and less frequently the differences in individuals' perception of sensory stimuli become the key of the segmentation of individuals to interpret the preferences and eating behaviors. I need to leave out some of these slides. We realize that the progress in research conducted in studying individual differences in responding to flavors, to tastes, and preferences, this has found a basis for its reasoning, and that is checking that there is a biological foundation, and this interacts or causes diversity in perception of food products, and this diversity determines different likings and therefore different preferences, and therefore it determines different consumptions. In fact, there are a series of indexes which have been tested, experimented, and we can segment the population according to their different reactions to food products. Something's happened to the slide here. It's not quite ordered, but these phenomena, at least the best known ones, are the prop status, which I will talk about shortly, the density of fungiform papillae, and thermal tasting, which determines people's ability to taste when there's a variation in temperature. A very quick variation takes place without stimulating of using substances, taste substances, and the hedonic value, and the classification in sweet likers and sweet dislikers, and there are variations of concentrations in sucrose. I will consider just two of these indexes, which can describe the different sensitivity to stimuli, because these are the most studied ones, and they're among the most interesting. The first is the prop status. That is to say, the different sensitivity to bitterness elicited by a couple of probyltherothyl and phenylthiocarbamide in aqueous solution. And for some individuals, this causes reactions which are extraordinary, and for others, nothing. This corresponds to genetic variations of a receptor gene, TAS2R38. This characteristic, for example, a proportion of non-tasters in the different world populations varied very greatly, as it's a phenotypic expression of this genetic variation. Why is this characteristic important? Well, it's because individuals this characteristic, which shows a different capacity to respond in terms of intensity to taste stimuli. It doesn't concern just the substances involved, but it can be extended to a whole series of substances. If you compare the response of caffeine and quinine to non-tasting individuals, the super tasters can perceive much more intensely this sensation than the other two. But extraordinarily, these individuals respond differently to real products. The characteristics that define the flavor of these real products. And it's easy to imagine that individuals who react differently develop different strategies in their consumption of these products. Just imagine that if you perceive bitterness very, very intensely, it'll be a person who uses more sugar to conceal this property, or will avoid consuming this product entirely. It's quite simple to understand the implications of this. If you think that the prop status in itself measures sensitivity not only to bitter or sour or sweet, but a whole series of tastes, whether it's tactile sensations, you will understand that this characteristic may be important for an understanding of why there's a development of diversified preferences. For example, being prop tasting is associated with creaminess, oiliness, or something which is, or viscosity, or something which is oral burning. And this is something which predicts the sensitivity to a series of stimuli which can be useful. But there's an extra step that we can take, that is to understand how these indexes are not linked just to the sensory perception, but also to food or eating behaviors. For example, for individuals who have a low response to prop, low score for prop, probably they'll consume more alcohol. Poor people who have this high alcohol consumption, we've mentioned them too often today. These are individuals who are less sensitive to increases in the intensity of sweetness or creaminess. They're less sensitive. Therefore, they should tend, to avoid less hedonistically sensitive to these fields. they tend to consume more vegetables than super taster individuals. There's a group of individuals who not only have a different perception of these in their sensory perceptions, but they link this perception to different likings, different behaviors. Similarly, depending on the studies, independently of the prop status, there's the effect of another index, which is defined by the number of papillae, the density of fungiform papillae. These are structures within which there are the taste buds. The higher the density of the taste buds, the greater should be the sensitivity to tastes. What happens if you take individuals who have got many papillae and individuals have few? You can split them halfway down, divide them in half, and there's a possibility that these individuals will respond differently in terms of intensity when these data are in aqueous solution, and when there's this diversity and this evaluation is made on real products. But above all, they have different likings. People with few papillae tend to prefer, in this case, for example, to develop less liking for coffee, with or without sugar. And why is this? It's simple. People who have higher papillae density, they perceive the bitterness of coffee, so they add more sugar. And this way, they develop a preference for coffee induced by the presence of sugar. But they increase their sugar consumption. So we go from a difference, a physiological difference, that is the number of the density of the papillae, which progresses to a food behavior, an eating behavior. Together with these facts, there are psychological factors which have their importance. These are not always considered as a particularly important fact in segmentation of consumers, whereas they should give information systematically. Food neophobia is the rejection of anything unfamiliar. The mechanism underlying this psychological trait is the fear of trying to taste a flavor that you don't like, and it's at its highest in individuals aged 2 to 6 years old. There's a genetic contribution. 25% of adults still have this food neophobia, moderate or severe, throughout their lives. And there's this tendency to generalize reduction in dietary variety. It's quite easy to measure using a scale that has been confirmed almost worldwide, this food neophobia scale. But it's interesting to note, a colleague, a doctor who's recently graduated, produced an interesting test that shows in a large number of children, 228 children, shows children aged 6 to 11, according to their level of neophobia, they like fruits and vegetables, and the liking for vegetables decreases strongly with this neophobia. And so this characteristic should be studied carefully to understand the reasons and to gradually try to overcome this because of the social importance that it has. In recent research that we carried out, it's a European product which we carried out here in collaboration with Bonduelle, what emerged was that the Italian adolescents, if divided between neophobic and neophilic, they respond in terms of liking of different products, different vegetables. They have different reactions. So we need to collect this information systematically so as to understand the best strategies to adopt to facilitate the consumption of healthy foods. I would like to stress that there is scientific evidence that makes it reasonable to think there's cause and effect behavior and the implications for society and industries. This is useful for development of food products and for improving conditions of nutrition in our societies. But we need to make some progress. We need to understand the impact of these individual differences in taste sensitivity, on food liking. We need to understand these mechanisms. They're not completely known to date. We need to develop methods which will be agreed upon and generalized, which will be validated, and that will be uniformly used in the scientific world. And we also need to understand the importance of sensitivity to food and liking and the effect it has on the conditions of well-being and health. And lastly, information must be disseminated to the general public, not just to the technical personnel. And the Italian Science of Sensory Science Association has the aim of exploring the individual differences in food preference among Italians. The Italian Society of Sensory Sciences has about 120 members present in 23 different laboratories in Italy. Some of these laboratories or these centers have taken the responsibility of collecting information in a 3-year period on a large number of individuals, between 3,000 to 5,000 people who will be recruited. This project will have the aim of clarifying the role of differences in understanding the sensitivity to food sensitivity and its role in developing preferences and food and eating behaviors, and to create a data set together with what is done in epidemiological studies to be useful for these people who produce food products and those who are responsible for defining policies to improve the well-being and the nutritional situation for citizens. And what we will do, it's not easy, but this is what we will do. We will have indices to measure sensitivity and these will be with crop status and the fungiform papillae density, and we will create a genome and the body mass index. It's not the best thing. Then we will collect information about socio-demographics and other information, gender, age, education, place of birth and residence, etc., certain consumption habits, and we will collect information about eating behavior and attitudes, and we will collect information about the lifestyle related to food. This doesn't concern only neophobia, which is perhaps the personality trait that's best known in the food field, but it's the question, the sensitivity to rewards and punishment, private body consciousness, and the disgust scale, and all of these can be connected to food likings. And the central theme will be collecting sensory data. This data in aqueous solution and in real products, we will measure the responsiveness to taste and mouth feel changes in real products, responses to salty, sweet, acid, sharp, etc. And we will also measure the variations in liking in order to be able to segment with different criteria our results. And we will have data, the stated preference liking for familiar and unfamiliar foods, and the choice, which produces alternatives in terms of taste and in terms of the nutritional content or health variants. I'm pleased to announce that after 8 months of existence of this project, in the recent weeks we are concluding the pilot phase of this study, this trial, which is quite a complex procedure, and soon we will start collecting data, and we are very happy that our proposal has been liked and has been successful with the Bonduelle Foundation.
Thank you, Erminio. We're running a little bit behind time. If somebody's got one pressing question to ask. We've always got time during lunch, of course, but I think we might break there. And please talk to Erminio during the lunch break about Italian Taste Project, or indeed the excellent data that he showed from his lab. And please join us back here at 2 o'clock. Thank you for your attention this morning. Thank you for your interest. And please thank once again Erminio and all the other speakers.